called Scientific Beekeeping. If you have not been there, shame on you. You need to go visit this webpage. There's more information there than you can possibly imagine. And I don't think he'll get it all in today, but he'll probably try to get most of it. So, if there are no more questions, Randy.
All right. Okay. Joe said okay on the video.
You can talk about Varroa if you want.
No, in the video, yeah. Okay, can you hear me? Okay. Interesting because this season I shifted away from nutrition and have been focusing on Varroa and just wrapping up some field trials just this week, just taking data 2 days ago on effective mite treatments. And I spoke 2 weeks ago in North Dakota about Varroa management and the future of the industry and where we're going. So one thing I don't like to do much is to do a cold presentation, a presentation that is just fresh. And I've been so busy with the bees that I threw this one together in the last couple of days, including a few hours this morning. So this will be the first time through on this presentation. So we'll see how it flows, okay? There'll be a lot of information. What I did is I started off with the biological basis of bee nutrition. And then I'm going to get into some technical aspects of mixing up pollen subs. First, is there interest in the technical aspects of mixing pollen subs? Because I'm going to skip that portion. There is, okay. And then after that, I'll get in. I'll go as long as we have time for. I just put in a bunch of field trials that I've run on bee nutrition. Most have not been published yet of how I'm actually testing things out and learning on this. So first, let's look at colony population dynamics. Let me see if this will turn. Oh, yeah. Now I don't have to turn my face away from me to see what's on the wall behind me. Those of you who've been following my articles know I've been focusing around this chart from Lloyd Harris's fantastic data set where he marked 100 bees in 35 colonies every 12 days with paint, and then every 12 days again would go back there, count how many of those bees were still surviving, mark another 100 bees, and measure the amount of sealed brood every 12 days in all those 35 colonies over the course of a year and a half. Monster, extremely valuable data set that sat for 20 years, not looked at, and he started digging it out a few years ago and publishing papers. I've been using it quite a bit for this. And what it shows is the age of shows a number of things. The dotted line, this is for Manitoba. Colonies started, this is a composite one. He started them from packages in the spring and then ran them through that entire year and till clear through to the next July or August when he killed off the colonies. I put the two together to make just 1 year's worth to show how this worked. The dotted line is the amount of brood, total brood, open and sealed brood in the colonies at one time. These are all averages. This is for Manitoba for colonies that went into a shed during the winter. The red area is the population of bees that are less than 12 days old at any time in the hive. So any time point. So at this time point, you had roughly 14,000 bees that were 0 to 12 days old. This orange is the number of bees, the next age group, next 12-day age group, up to 24 days, up to 36 days, up to 48. Very few bees during the summer live to 60 days. The numbers along here is the average age of all the worker bees in the hive at that time point. And what you can see is from early spring until late summer, the average age never gets over 26 days. Sometimes it's younger than that. So you have a very young population of bees at that time of year. During the winter, if you have a cold winter area, as you do here, the bees start to age, the surviving bees after the fall turnover, when all the bees that have reared brood or foraged fly off, abandon the hive and die. You start getting bees aging, where you get bees that are living up to 200, 300 days of age until the spring turnover, when these aged bees in their last gasp of energy raise the huge new cohort of bees. At this point, you can have 3 times as many bees in the brood as you have adult bees covering them. That's that critical spring turnover, when suddenly then it will shift over here. And if the colony gets through this spring turnover, this critical point right here, then it just takes off in this linear growth phase. All this stuff is on my website. the point of all this, I tracked this out for 2 years here, is that actual colony population is a function of how much pollen is coming in. How much the nutrition of a hive is mainly pollen. That's where the protein comes from. Now, we could just erase all these dates and use this anywhere in the world. I just got back recently from speaking in Mexico, and we talk about winter because winter correlates with cold weather and short days here. To the bees, winter correlates with pollen availability. And in Mexico, in areas where it's quite hot during the summer, the bees, their fall comes in July, and their winter starts in August, and the bees just shut down, stop rearing brood, and go into what looks like a wintering condition. They don't have to form the cluster. And their spring starts in November when it starts to rain again. So to the bee colony, the winter-summer demographics are more about pollen availability than they are for cold. In California, you would take this graph where I live, and you just cut off this end, and just stretch this out. If you're further up in Canada, you would squeeze this together, and this part would be longer. So this can be adjusted for any area. And again, it's just all about pollen. What I did here is I tracked the amount of pollen availability where I live, where we started off early in January with the alder trees start to bloom. We get a little bit of spring bloom. Almond pollination has a huge amount of pollen. Then we have the fruit bloom after that, and then after the fruit bloom, when the apples and cherries are done blooming, we have about 2 weeks with almost nothing coming in. Then it starts to build up on the wildflowers. By midsummer, it peaks out, drops to almost nothing. And then right now we're enjoying just a little tiny bit of fall bloom. In California, we don't get the fall bloom that you get east of the Rockies, where you have rain during the summer, or rain at all. Luckily, we got some thunderstorms a couple of days ago, and you could hear the sigh of relief from all the vegetation around me. So let's look at the protein flow in the hive. So just to get the basics of the biology, here's the dynamics of the hive. You have an energy reserve. The honeybees are all about honey, about storing energy. Just like we may store energy for the winter in the form of coal or the propane or heating oil, they store it in the form of honey. So this is your energy reserve, the sealed honey. Then you have an energy interface, which is right between the bee bread and the honey. The energy interface is where they're either uncapping stored honey and utilizing that as an energy source, or they are depositing fresh nectar as they turn it into honey for storage. So when the colony has a surplus of nectar coming in, the brood nest gets pushed down. When the bees are consuming their honey to stay warm or for energy for flying, they eat their way up here and they'll move the brood nest up. So it's a dynamic area right here. The protein interface is right here, right between the brood and the honey. This is where you have this rapid turnover of incoming pollen that is quickly consumed by the nurse bees, and if converted into, to protect that food, to put it into their bodies as fast as they can. Pollen is very easily attacked by microbes or decomposers. Insects, everything wants to eat pollen. Pollen is very nutritious. So the first thing the bees do is they hide it inside their bodies. They get it into their gut, they put it into their fat bodies, they put it into hypopharyngeal glands, and they put it into the brood. And they use the brood as a protein reserve that they can cannibalize at any point and take protein back out of. If there's a surplus of pollen still left after they've eaten all that they can that day, then they go in the process of fermenting it by packing it down with their heads, adding some nectar to it, putting a thin film of honey over the top so it forms an anaerobic with that oxygen substance there. And we're going to talk about that later, what happens then for long-term storage. Then you have your protein and energy demand, which is the brood right here and the workers, and for many of us, our food base for raising varroa right here inside the sealed brood. So I pointed out last night, many of the groups of the non-professional beekeepers that I talked to really should more be termed as varroa farmers than beekeepers. And they do a really good job of just growing lots of varroa food every year. Now, the neat thing that bees do is they do something similar to what mammals do. Female mammals do not feed their babies raw food. The mother eats the food, digests it, and then secretes a perfectly nutritious food for the baby. So we call it milk. The bees do a very similar thing. They produce a substance called jelly, and that is the food not only for their babies, but here's the neat thing. All species use a protein, a yolk precursor called vitellogenin. Vitello means yolk, genin means creation. So it creates yolk out of this. In the honeybee, most of the females don't lay eggs. They have no real use for that vitellogenin, so they give it another use, and they use it as a storage protein and to put into the jelly and use it for protein transfer throughout the hive. The way this works is the only bees in the hive that are able to digest pollen and get the protein out of it are the nurse bees. The bees typically from about 2 days old after they emerge to about 15 days old. And that's not written in stone. That can be extended or shortened up. Once the bees transition into older duties, going from mid-age bees into foragers, they shift their digestive enzymes and they lose their ability to digest pollen completely. So if a forager bee happens to eat some pollen, it just shoots through its gut in a few hours without any digestion taking place at all. So it's feeding foragers pollen is of no benefit to them. So what they have to do is they have to beg for a little bit of jelly from the nurse bees. And you can check this really easy. If you're doing nosema sampling, we're using the Ziploc method of putting some bees into a bag, crushing them, and adding water to check for nosema spores. If you've got a bunch of bees from the brood nest, the liquid around will look like this, full of pollen that's coming from the guts of the nurse bees. If you do it with foragers, the liquid will be clear because there's almost no pollen in forager guts. Very easy just to age the bees by crushing them, putting in some water, and looking at the color. It'll just tell you whether they were forager bees or nurse bees. So we have this transition of the incoming pollen or the stored pollen, then shifting for recruitment of rearing new bees in the colony. And here's how the flow works. From the foragers bringing it in to the temporary storage here, which can be very quick. It can be eaten within hours, or it can be stored as bee bread for months. The nurse bees then producing the jelly, they pass it among themselves in a colony communal stomach. So if there's jelly abundant in the colony, it's spread throughout the whole colony. Everybody suffers or benefits the same. The whole colony knows. All the foragers know. Everybody knows what the protein condition is in the colony at any moment, because those foragers coming back beg for some jelly from the nurse bees, and if they don't get much jelly, that tells the foragers, hey, we're short on protein, we better go start foraging for pollen. If there's abundant jelly, that says, oh, well, protein's not that important right now, we can forage for nectar instead. All these feedback loops in the colonies for the regulation of the foraging are just fascinating. And then from the nurses, then that jelly goes out to all the rest of the bees in the hive. The nurse bees, their hypopharyngeal glands are fully developed. They're analogous to the mammary glands of mammals. To produce that jelly, they atrophy when they become foragers and no longer produce jelly. The foragers are able to reverse this and reactivate their hypopharyngeal glands. They're not quite as good as they were when they're nurse bees, but if you were to start say packaged bees with nothing but just foragers, they would be able to go back and start producing jelly and feeding the larvae. The vitellogenin is also stored in the fat bodies. These are 2 bee abdomens here. I got a new pointer. Dr. Eric Musson pointed out at a recent conference that some % of the male population is unable to see red. And I had a red pointer, and I said, oh man, I don't want people not to be able to see what I'm pointing at, so I bought this green pointer. And I'll tell you, we went out on our deck, which overlooks a mountain range, and went out at night. We couldn't wait. It kept getting darker and darker. We kept going out and trying it, my wife and I, and with our binoculars to look at the tree trunks across the canyon. And you can pick this little pointer up clear across the canyon at night. It's just amazing how powerful these things are. If you don't believe me, raise your hand and I'll get you in the eyes right now. Anyway, these are the fat bodies stored. So this would be a nurse bee or a winter bee, or diutinous bee or survivor bee. So the bees, I was talking to Steve Coy this morning about the Russian bees. And in my area during the summer when there's very little pollen, late summer, they go into survivor mode, the diutinous mode, or similar to winter bees, where they stop rearing brood completely, and they just load up their abdomens with all the protein that they can then hold, and they just hunker down, they wait for things to get better, okay? So this is what these diutinous or winter bees' abdomens would look like. Now, the problem when vitellogenin levels get low in a colony is it really changes the bees a lot. They start to age more quickly, start to forage more quickly, they get sick much easier, they can't resist the viruses, and they shut down their brood rearing. This has huge effects on the colony dynamics, colony health and colony growth. And it happens very, very fast. If you get even a day's rain, Karl Kreilsheim in Germany found that the nurses cut back immediately on the amount of jelly that they feed to the larvae. Now, the larvae are only fed for 6 days. You get cut back on jelly for 1 day, those bees, when they emerge as foragers, as workers later on, will not be the foragers that they could have been if they had been fed continuously for all those 6 days. So it's amazing how quickly the nurses respond and start cutting back on the amount of jelly coming in. Protein requirements. The colony curve, population curve, is based on just 2 things, recruitment and attrition. Recruitment is the number of young bees emerging each day, and attrition is the number of bees that are dying, either from just aging or wearing out or whatever, or altruistic self-removal. Anytime a bee feels sick due to exposure to a toxin, due to a virus infection, due to a nosema infection, that bee, in order to protect its sisters, sacrifices itself. It just flies out of the hive and it doesn't come back. So a large part of this colony nutrition is based upon this altruistic self-removal. In the most extreme case of altruistic self-removal, we call that colony collapse disorder. And if you never watched a colony evaporate in 2 days, it's pretty damn impressive how a big, strong colony, if it has a combination of, say, a paralytic virus and nosema infection and a little nutritional stress, can just go poof, and the bees just all fly out, leaving the brood behind. It's amazing. We went through it pretty bad in California around 2000, from 2004 to about 2008. We had a lot of sudden colony collapses. Concurrent with the invasion of Nosema ceranae, I kind of have this feeling that Nosema ceranae had a lot to do with that colony collapse, at least where we lived in California. You can see here what I plotted out here for just a typical colony growth curve over the season. This would be your daily change in number of bees in the hive. So you're gaining 300 bees per day. So like if a queen's laying 1,500 eggs, and say you have 100% survival of those, you'd have 1,500 new bees being recruited that day. If you have a net gain of 300, that means that 1,200 bees are dying from attrition on that day. Now the queen's going to lay at about the same rate, so this is saying up here, you're having fewer, remember the colony demographics? We're getting a younger colony right here. Now you have fewer bees die, so more bees can survive, so now we're getting a gain. And at fastest colony growth with a really good queen, about that 600 bees per day gain is about the best you're going to get. And that's why you get that linear growth, because it's a constant. It's not exponential, it's just a constant. You're gaining that many bees per day. The colony tops out usually around July for the honey flow, and then it starts to lose bees at a high rate and then at a lower rate into the winter. The recruitment to maintain, or to build or to maintain that rate is all dependent mainly upon the queen's egg laying rate. The queen's egg laying rate determines what your top out population will be. The colony will grow at that rate until you reach an equilibrium. Lloyd Harris figured out what the survivability curve was for the worker bees, and at some point, when the colony grows to a certain point, the natural attrition of the workers will match the egg-laying rate of the queen. And at that point, the colony just stops growing. That's maximum top-out population. You cannot make the colony grow any bigger than that. It takes about 60 days from when the brood nest is established till the colony reaches maximum population. And that maximum population is set by the queen's egg-laying ability. If she can only lay 1,000 eggs a day, compared to a queen that can lay 2,000 eggs a day, the maximum population is going to be about 35,000 for a queen laying 1,000 eggs a day. You can get up to 70,000 or so for a queen that can lay 2,000 eggs a day. So that's set by the queen's egg-laying rate. And it's limited by the amount of protein coming in. So the colony that, if it rains several days a week, will not be able to grow to the extent that a colony that has that can forage every day on good pollen can grow. If anybody has any questions at any point, feel free to shoot a hand up too.
How long does it take to build that vitellogen level in those winter bees? Is there a durational period, or can it happen?
It's very quick if it's good nutrition. Yeah, as soon as they digest it, it's right into vitellogen right then. So it could considerably happen like in a day or so. Okay, colony growth. So this is a picture I took. What we do in the springtime, and beekeeping is different everywhere you go. So I don't try to make any generalities about beekeeping. My experience is all in the California foothills of beekeeping. I can talk at length about beekeeping in the California foothills. What we do is we come back from almonds and we split every colony we own. We wind up with 4 colonies for every one that we bring back from the almonds. We sell about a third or a half of our operation off every spring as nukes, and then we rebuild the rest to up for the honey flow. So this would be in early spring after we'd split our colonies into 5-frame nukes, and after a few weeks they have built up and they look like this. These colonies are living hand to mouth right now. They're just solid brood in here, but we have a real dependable nectar and pollen flow at that time of year, and we can super them up and get ready for the honey flow right here. The amazing thing is just how quickly they can grow. So to put it in perspective, I looked at the growth rate of the fastest growing animal I could think of, and that'd be the hybrid broiler chicken. When I was in my early 20s, I ran a feed store, and I read Morrison's Feeds and Feeding, which was the classic animal husbandry book at that time, and it took 19 weeks to grow a broiler chicken for harvest from egg to harvest, 19 weeks. Who can tell us how long it takes to grow a broiler chicken from egg to harvest now? 6 weeks. That's the progress we have made in selective breeding of the chickens, okay? We haven't made any near that kind of progress with the honeybee. So these hybrid broiler chickens are an incredibly fast-growing animal, and it occurred to me they pass the 2-pound mark along that growth, which is the same as a 2-pound package of bees. And I can compare the growth rates between a 2-pound package of bees and a 2-pound broiler chicken. So I plotted this out by days right here and by weight in pounds. So here's the broiler chicken growth rate. Now I found 2 data sets for honeybee growth rate for the weight of the actual bees in a package. Now in a package, it's going to drop for 21 days because the bees die before you get any new brood. And then it starts to grow. And once it starts to grow, they grow faster than the broiler chicken. So the fastest growing livestock we have gets left in the dust by the honeybees. But this doesn't even begin to tell the whole story. Because the broiler chicken, if it's putting on any extra food, it stores it as fat under its skin, on its body. Okay, you've all seen the fat there. That's the reserve of the broiler chicken. These 2 lines for the honeybees don't count their reserves, because where do they store their reserves? In the comb, in the hive. So let's look at the actual weight of the total bees. So here's the broiler chicken just slowly slogging along here. This is what a bee colony does right here. It has a rate of growth that we can't even conceive when we compare it to other livestock. If you're growing a livestock that fast, what's that tell you about nutritional needs? They're beyond, way beyond any other kind of livestock. Bees need to get maximum productivity out of a colony, you need to have absolutely optimal nutrition with 0 breaks at any point, okay? Any break, even for a day, will slow that colony down on its buildup. Everybody clear on this? Okay, just for growth. Now, I'm not even talking about disease resistance right now, or varroa resistance, or virus resistance. When I do calc it out based on Lloyd's survivorship data, it works out that if you walk out to a hive and people say, oh, yeah, my bees get to know me after a while. Well, not if you only visit them every 42 days, because it's a whole different population of bees every 42 days. Total turnover of that population of bees. That's how long it takes to turn them over. And bees are made out of protein. So to replace all those bees in the hive, you need to have enough protein to turn over the population in 42 days. And for a decent-sized colony, that takes about 1 to 2 pounds of high-quality pollen per week, every week. And if you get a week's worth of rain and no pollen comes in, that means you got to double your pollen income the next week to make up for that, to take care of the deficiency that they went into. That amount of pollen works out to be about 100 to 200 grams of protein per week. That is for colony maintenance, not for colony growth. For colony growth, you need even more than that. So those of you who are taking notes on numbers, these are your numbers of, if you're looking at supplemental feeding, 100, 200 grams to maintain a decent-sized colony, even more of protein to grow a colony.
Excuse me, are you saying that's for the winter, our winter as well?
Not winter. This is for during the summer. During the summer. During the winter, the colony's not growing, so it needs very little protein. Okay, unless you're going to almond pollination and you want to grow it, then it does need protein. Does that make sense?
No, I just want to check, about storage for the fall, for the winter, regardless.
Yeah, unless bees are recruiting by rearing new bees, they don't need much protein at all. It's only when they're recruiting, when they're raising brood. Everybody good on that? Okay, to get that amount of protein requires, again, 1 to 2 pounds of pollen or bee bread coming in. As soon as it's reduced, they reduce brood rearing. They cut back on the brood rearing, or they start cannibalizing the eggs of the brood. I don't know how many people have noticed this. In my area in the springtime, especially during our pollen dearth after the fruit bloom, those queens are just laying their hearts out every single day, and the workers just follow them behind and eat those eggs just as fast as the queen lays them. And they'll keep consuming those eggs until pollen starts coming back in, and then they'll start feeding the larvae again. So you go out every day and you go, wow, that queen's just kicking butt, look at all these eggs. But the colony's not growing. Why is the colony not growing? And if you look carefully, you'll see that the queens, the workers, the nurses, eat the eggs right back up as fast as the queens lay them. So it's not just the queen that sets the egg-laying rate in the colony. It's the nurse bees who decide which larvae you're going to allow to live and they're going to feed. Yeah?
Does it matter which strain of nurse bees you have, Russian versus Italian versus any of the other?
I couldn't tell you that, but I would doubt it. But I can't tell you, I don't have any doubt on that.
Russians will shut down in your dearth, so that
Yeah.
means there's no pollen. So who decides that, the queen or
The Italian bees, our commercial Italian bees, are bred not to shut down.
how about Russians?
The Russians are, we have a Russian expert right here.
The Russians are more, I've seen that happen more often when you just described about them eating a brood back and stuff.
Uh-huh.
Russian colonies. Russians are more,
Environmentally sensitive,
Yes.
or responsive, environmentally responsive.
yes.
Yeah. So the commercial Italian stocks we breed are bred not to be environmentally responsive. They're bred to just be full tilt going all the time, and that's why it's very easy for them to fail if they don't have adequate protein coming in. Okay? And that's part of our breeding selection for that. Okay? The guys who breed the Italian stocks are package producers. And what do you want with package production? You want monster bees that just brood up and just keep going and keep going, despite what's ever happening in the environment. So we breed for bees that will do that. If the nectar or the pollen flow and nectar flow gets reduced by rain seriously, this is a picture I took. We had a rainfall event in April a few years ago where the bees couldn't, or snow and rain came in right when those colonies looked like that picture I showed you with the single boxes, wall-to-wall bees, and it snowed on them. It wasn't that cold, but they could not forage. And those colonies were all living hand to mouth. They were fairly light. We didn't expect that snow at that point. And immediately the bees started responding. This has taken 5 days after that snowfall event. And you can see the bees have consumed every bit of honey in the colony, every bit of bee bread. Then they eat the eggs, then they eat the young larvae, then the older larvae. They don't eat the sealed pupae generally because they don't need any more feeding. So that's the last salvation for the colony, is to have those sealed pupae emerge. So they leave the sealed pupae in there, but they eat everything up. Those colonies, we got out during that weather event and that we were able to give a little bit of feed to, just cooked right along and made a honey crop. Those that cannibalized their brood, they didn't die, most of them, but then had to rebuild after that. None of them put on enough honey to make it through the winter. We had to supplementally feed them. So just an ounce of prevention worth a pound to cure. The ounce of prevention of going out during that rainy weather and putting a little bit of feed on the colonies allowed the colonies for the rest of the year to take care of themselves. Those that we allowed that they got to this point did not take care of themselves.
You said feed. Are you talking about syrup or pollen?
We put both on. Yeah. Nosema, this is a picture of a bee's tracheal tube here. All these little bright spots are nosema spores. That's how dense nosema gets into the bee's gut. Nosema, to grow this population of nosema, it takes a lot of protein, and the nosema steals protein from the bee's gut, and nosema does not have ATP. It steals the energy directly from the bees. So these guys just suck up energy and protein out of the bee's gut, so they hamper the bee's ability. And again, the poor nutrition leads to precocious foraging. So instead of waiting until they're 15 days old to start foraging, they may start foraging when they're 4 days old or 6 days old. The time clock for a bee's lifespan pretty much starts when they start foraging. They're only going to forage for a week or so. So that means if they start foraging 5 or 6 days earlier, they're going to have that knocked off the end of their lifespan. So what that does is on your, I wonder if I put, yeah, on this curve right here, it's going to reduce this number if they're going uphill, or they're going to increase this number if they're going to go downhill, and the colony will be much poorer for that early foraging. This is part of what happens in CCD. As the bees start to evaporate with their altruistic self-removal, it forces the younger bees to start foraging earlier, and you get this positive feedback loop that they stop feeding brood, they start dying quicker, and just in a few days it snowballs and the colony population evaporates. I have an article called Sick Bees 2 that has the model that I developed for what happens in colony collapse. You're welcome. It's on my website, easy to get to. Another study by Gloria Degrade Hoffman showed that bees, when they emerge, pretty much every bee that is laid as an egg now has deformed wing virus, vertically transmitted from the mother. And when they emerge as larvae, if they get good nutrition, they suppress that virus and keep it in the latent state. So pretty much every bee has deforming virus all the time. Just like everybody in this room has herpes virus. Most of you this age probably have chickenpox virus. You keep them latent in your body. Unless your immune system is suppressed, if you get AIDS or you get old, then those suppressed viruses can flare back up. What she found was critical was how much protein those larvae were fed. They're able to keep that virus down. And the ones that were fed pollen kept the virus level very, very low. The ones that were fed a pollen virus level went up more, and the ones that were fed no pollen went up quite a bit. And that ties right into this, that poor protein really exacerbates varroa problems. What I found in the Sierra foothills, I just couldn't keep bees alive during the summer in the Sierra foothills, so I would transport all my bees up to irrigated pasture in Nevada. I did that for many years, simply because they would survive up there. On good forage, the bees that go up out of California to North Dakota to Montana to irrigated pasture elsewhere just do way better than the bees that are left in California. What we've done, my sons and I have done, and this is what was my interest in bee nutrition, we learned how to feed the bees supplemental protein, and now we keep them all near home. They're still not as good as if we would have taken them to good pasture, but we're able to keep them. It's a lifestyle choice for us to stay near home rather than being migratory beekeepers.
Well, if you don't mind, how big is your operation? I just want to get the picture.
We run about 1,200 hives. So for California-wise, we consider us very small beekeepers, just slightly above hobby level.
So how many people operate it?
Me and my 2 sons. Okay, now this brings up another question, and that's bee bread and microbes, or the microbial community that's within the bee, and especially the relationship of that and the fermentation of pollen. A very compelling story has grown over the years that the microbes are involved in improving the nutritional quality of the pollen in the process of fermentation. And then every time that story gets retold, it got better and better. And then in the internet in recent years, I heard, well, bees can't even digest pollen. It has to be fermented to get any nutritional value out of it. So that's the extreme version of that right there. So the question is this fermentation of pollen into bee bread really necessary for digestion, or is it simply for preservation? We're learning a lot about bees since we started getting elevated colony mortality. As the virus has evolved, as varroa settled in, and as Nosema ceranae invaded most of the world around the year 2000, suddenly it became much harder for bees to to stay alive. And what you can see is right around then, 2004 is when we had our first major shortage of bees for almond pollination in California, brought it to the attention. Dave Hackenberg, 2007, then made all the papers, and all the public became aware of colony collapse disorder. And suddenly it was a gold mine of funding for anybody who wanted to be a bee researcher. You just put CCD in your proposal and people threw money at you. Just like now, if you just put neonicotinoids in it, people just throw money at you. And this is the number of publications, scientific publications with the words Apis mellifera on them over time. And you can see research on bees just exploded following CCD. So we've learned a lot about bees in this period of time. The ARS lab just did something very wise. They do this, is you bring in a researcher who knows nothing about bees, but knows a whole lot about something else. And you say, look at bee issues with fresh eyes that you haven't read all the bee books and got all the wrong information, okay? And there's been a number of these guys come in, and it's fantastic because these guys come in and say, well, Kirk might say this, but most of them are a little more discreet. They say, you idiots, you've been doing it all wrong. And I see this over and over again, that the research community will publish all these papers based upon sloppy methodology or something. You bring somebody else in from outside and said, no, you guys screwed up. You might as well just retract all those papers because that was all wrong findings. But now they're all in the bee books and in the public knowledge base. So Kirk is one of those guys. And Kirk and I hit it off, and we communicate constantly on this. I just published this chart. This is a summary of the work from Kirk, from Nancy Moran, from Martinson, from all the people who are using next generation sequencing to figure out what microbes are in the bee gut. Instead of using culture-dependent sequencing, where you make petri dishes and you try to grow bacteria and name them, you only see maybe 1/10 or 1/100th of the species when you do that. If you use next-generation sequencing, where you actually look at the DNA in there, then what you come up with, you don't come up with species, you come up with OTUs. And so when you read these things, you'll see how many OTUs. An OTU is an operational taxonomic unit, which means it's a species, but there's no name for it yet. When they finally figure out, when they culture that OTU and then can extract the DNA and say, oh yes, this is a species, now we can name it, then it gets a species name. When they started with next generation sequencing, they found out that we don't have names for most all of the bacteria out there, and those found in bees. There are a few that have developed some kind of symbiotic relationship, mainly in the bee hindgut, that digest the undigested exines of the pollen coat and the other waste materials in the hindgut. There's a few species that have make a biofilm inside the ileum, which is between the stomach and the rectum right here. And then the crop right here and the midgut are very inhospitable to bacteria. Very few bacteria in either the crop or the true stomach. And the reason for this is that the nectar coming in is just full of microbes. It's full of yeast fermenting, it's full of bacteria, full of a lot of things that would rapidly degrade that nectar before it could be made into honey, or actually make the bees get sick. So when the foragers take the nectar into their crop, they want to kill most all the bacteria and the yeast in the crop. And then when they pass it to the mid-age bees in the hive, they continue that process, filtering out anything that's in there with their proventricular valve. And then when they disgorge that, it comes out cleaner, more microbe-free. There also are a couple of them that live in the hypopharyngeal glands that are adapted for the high osmotic honey and jelly. And so these two species right here, Lactobacillus kunki and Parasaccharobacter, this was an OTU until last year. Now it has a name, the name by Kirk Anderson's lab. Both of these, this one is found in the jelly, inside the hypopharyngeal glands, very well adapted with the bees, and confers immunity to the larvae, helps to clean up honey, and helps in the process of preserving the bee bread. Also, the Lactobacillus kunki is the main one that's involved in bee bread. But it's not, these two do not come from the bee's gut at all. They come from either the environment, from the flowers, this one here, or are passed from the nurse bees to the larvae by feeding. So let's look at this process of the transition of fresh pollen right here to the bee bread, where they pack the air out and they put a little bit of honey over there that allow it to ferment. The pollen is brought in by the foragers, scraped off their legs, loose into the cells. The forager leaves. A mid-age bee comes in. The foragers already added some nectar to make it stick to their legs if they need to. The mid-age bee adds more nectar to that and takes their head and packs it down, packs all the oxygen out of there, and puts it down in layers. So it may be one species, it may be several species of pollen in there, and then caps it over with honey to exclude or slow down the transfer of oxygen. And then things start happening here, this fermentation process. This is data from Kirk. These are the number of bacteria per gram of bee bread over time, from fresh coming in, less than 24 hours, and up to 96 hours. And what you can see is the bacteria, mainly Lactobacillus conchii, just explode in the first 2 days in there. Lactobacillus conchii feeds on the fructose, doesn't feed on pollen at all. It feeds on the fructose that the nurse bees add to the pollen and converts the fructose into lactic acid. The lactic acid kills all of its competitors and eventually shuts down Lactobacillus conchii. It goes into a dormant state, and now it just waits in this state in this bee bread. When the bees later then take that bee bread into their crop, add fresh nectar to it, it dilutes it, the Lactobacillus conchii can be reactivated and ready to start growing again. So this is all part of this processing of the bees using the lactic acid fermentation, the high sugar content, and the addition of preservatives, the glucose oxidase from their saliva. Same that we do for making jelly or pickles or that. It's the same kind of biological way of preserving foods. So we got nothing on the bees. They were doing it long before humans were learning how to preserve foods. But you can see this is the bacteria then drop off by 96 hours to almost nothing in there. Not ever being willing to believe anybody at face value, I checked things out myself. So I took bee bread and put it under a scope and looked at it, and sure enough, in mature bee bread, there's almost no fungi or bacteria on it all. You don't have to take my word for it. It's really easy to do with a scope. You just go out and take a look at it. Kirk sent me a scanning electron micrograph of this. The orange or the pink stuff is dried. You have to dry things to do a electron micrograph. So this is the dried nectar, the sugars that are in there, but you can see there's no bacteria growing on this pollen right here, as opposed to this, which is taken from a bee's hindgut, and you can see the bacteria growing all over the pollen grains. So in bee bread, you don't have the bacteria growing on it. You do on the hindgut. Yeah, Steve?
You mentioned fungi.
Hang on. You got to have just a moment's patience, okay? But I'm glad you're paying attention. Now, the second thing that Kirk looked at is, well, if the digestion of pollen was dependent upon the fermentation, then the nurse bees would prefer to eat aged pollen that was fully digested. So they went out to a whole bunch of colonies, pulled out one frame from each colony at over 3 months, and they repeated again, lots of replicas. Kirk is a really good scientist. And they put a plastic sheet over each frame and marked every frame with pollen in it, every cell with pollen in it, and would go back every day, and they could track the consumption of that pollen over time. And what they found is that most of the pollen in these colonies at the time of year was in the old stored pollen. But as far as relative consumption, the bees much preferred to eat the fresh pollen. He couldn't tell about the first day's incoming pollen because there's no time to mark it. So he doesn't know how much of the incoming pollen is eaten in the first day before it ever starts fermenting. My guess is it's probably quite a bit. But as the pollen aged, it became less and less attractive to the nurse bee. So this did not support the digestion hypothesis either. And again, I was curious about that. So I took some bee bread and diluted it with a little bit of sugar syrup and put it on my kitchen counter for 10 days and let it just ferment like crazy. And sure enough, all these grayish things, these are yeasts, just growing like crazy inside there. So man, the yeast just grow like crazy. And here's some other, this actually is not, I'm not sure if this is pollen or, no, I guess it is. Yeah, that is another pollen. But look at the yeast just growing like crazy in that fermented pollen right there. So Kirk then looked at the yeast. This is the yeast abundance over time, 0 to 24 hours, 24, 28, and up to 96. And the yeasts also spike along with Lactobacillus conchii until the lactic acid suppresses them. Notice that this completely overlaps with the bee preference for consumption. Bees, apparently, the nurse bees kind of like that yeasty taste or smell. So Kirk took the next step and he analyzed the vapors coming off of the fermented bee bread. This, I don't think this has been published yet by anybody. So this is 1 to 2 day old, and he reversed these two spikes from the gas chromatograph here. So a spike means as you put this material through the machine, it heats it up, and then it has to go through a medium, and then as each chemical comes out, it separates them into each different chemical. So each one of these spikes is some chemical odor coming out. So for the fresh stored pollen, you see there's a lot of spikes at 1 to 2 day old. When the pollen's older, there's much less of the aroma. So imagine those of you who have ever made beer or fresh fermented pickles, you know that fermentation smell kind of makes your mouth water. Well, apparently it does the same thing for the nurse bees too. So it could be a nutritional benefit from the yeasts eating the sugars in the bee bread, but not the protein. They're not getting the protein out of there. I'm going to come to that in a second. So I'm not saying, and Kirk's not saying that there's no nutritional benefit from the fermentation, but that it's not necessary to get the protein out of there. And so then I did another test also, and Kirk did this also, but I think my photographs were better. So these are pollen grains after I left it sit on the kitchen counter fermenting for 10 days. And what you notice, those pollen grains are completely intact. All the protein is still inside them after 10 days of vigorous fermentation on the kitchen. To compare that with the pollen grains after they've been in a nurse bee, gone through the nurse bee's digestive tract, and most all of them are emptied out. So nurse bees can very easily, within a number of hours, completely digest the pollen. Bacteria, given 10 days with all of fermentation, can't touch them. Okay? So the end result was, it's not for nutrition, it's for preservation. Okay? So those of you who have spent a lot of, I don't know if anybody in this room has, but a number of beekeepers have spent a lot of time and effort fermenting artificial diets to try to improve them. I'm not sure how much more effort should be spent on that.